before I go to the genetic diversity, I see it like I compare the very inbred population with a map population. So you have a pure Mexican versus a Mexican and Guatemala, or versus a TREC hybrid Mexican Guatemalan. So you really a mixed population like Mexican Guatemala and West India will be equivalent to your puppy who have like 3 or 4 or 10 races, and your only pure will be like an inbred population like these dogs. So because they are fixed and they are all lack of genetic diversity, like the very expensive pedigree dogs, they don't have a lot of problems. I mean, you have these dogs, you have to take them to the vet, you have more diseases. But if you have a dog who has a mix of everything, that dog is a mule. It's very strong, doesn't get sick, and they can withstand a lot of things. So that's the idea that I have for the product. Looking the perfect combinations of these who can get me the best rootstock that can work for disease and for abiotic stressors. Now, this is the genetic diversity of our young class. So likely when I took the program, I was scared what I was going to see, but I was happy that looking the genetic, we have all our foundation. So we have pure races, we have 2 hybrids, all the 2 hybrid races, and we have the mixed population. So we have what to work with. If I will have only Mexicans, then I will be in a problem because then I will need to bring material to enrich the other races. So why we look for the rootstock? Because it's the foundation of the tree. Now people tell me sometimes, why you're looking laurel in the scion, or why you're looking Botrosphaeria in the scion? Because the rootstock, there is a rootstock scion interaction. So you connect the vascular system, and through this vascular system, you have a lot of interchange. Let's see, in the case of the resistant, which is what I work with, if you have a very resistant rootstock against Phytophthora, there is a mechanism of plant immunity that they are shared. And some of them are based in hormone signals like salicylic acid, or small peptide proteins that enhance resistance, or other things that they can go through the vascular system. So it can, but these susceptible scion, they don't have the ability because they don't have that strong immune system. So this big rootstock can produce this small component that is highly resistant, maybe more salicylic acid, maybe a very important peptide. And it can translate that peptide and go above the ground and make it more resistant. And the same has been found for salinity and for G. So when I was making my presentation, I would like to explain a little bit on the rootstocks. What the type of rootstocks, because we have 2 types, the seedling rootstocks and the clonal rootstocks. So there are only a few countries, and in US, a few states like California, who use clonal rootstocks, and I'm going to explain that in a minute. But the majority of the avocado world uses seedlings. Okay, the only country that is using clonal completely is South Africa and Chile, for example. But they are getting or moving into clonal, but the majority use seedling rootstock. Why? One of the main explanations is the numbers and the money-wise. It's way cheaper. You produce your seedlings or buy your seeds and establish your orchard, okay? Well, in the clonals, Broca can sell you a clonal tree for $55. Other nurseries can sell you a microclonal, which I'm going to explain later, for a cheaper price, maybe not 50, but 10 or something, 20, a little bit cheap. And then hopefully we can have tissue culture that you can harvest for $15 or $20, who knows? But in the seedlings, you don't have to invest. Which is the problem, is that the seedlings come from a sexual reproduction, okay? You can have a selfing or you can have a cross. Selfing meaning that it's the mother who self, and it will produce the progeny without the parent or another genotype. When you have a cross, you have a mommy and a daddy, that they will be different individuals. You will still have crossing over. That's what when people say that asexual reproduction or selfing are identical and clonal, that's not true. Because you go back to genetics, asexual, your reproduction in selfing, you also have a crossing over. So you will have some kind of reshuffling in the genome, so it won't be completely clonal. In the case of the clonal, you will see that it's completely exactly like the original mother. because you reproduce it completely like vegetative. You take the badwood and you multiply the badwood from one tree, vegetative, and they will be exactly the same color. When you do a seedling, some of them will be similar to the mother, but some of them will be totally different. Will be a small crappie, will be other color, different morphology, because there are seedlings, either from self or outcross. If you do self, there will be genetic variation, but it will be less than outcross. And within outcross, you can have seedlings who are half sister and brother, or seedlings which are called half seeds, or you will have seedlings that came from the same mommy and daddy, which we call them full seeds. Okay. In the case of the clonal types, we have, that's depending on the technique to do the clonal trees. You can have clonals with frolic method that I'm going to kind of like work out later, microclonals and tissue culture. In the case of the tissue culture, you also can have somatic variation, but it's less. And it depends, I guess, in the variety, but nobody knows because it's very hard to tissue culture in avocado. If we wanted to, well, just to think, looking at that, the program knows that we need both. We need material who can use as a seedling, a material that it will use as a cloner. So what we're looking in the program is trying to find a rootstock who have good characteristics, good resistance to abiotic, to pathogens, and to water stress, for example, but with the level of selfing that is higher. And I will explain that later. There are different varieties, different rootstocks, and some of them can be completely self, have 100% of selfing, and then some of them can have 10% of selfing. So if you want to use a rootstock for seedling, it's better to use something that has a low probability of outcrossing, which is available to find in a jar plant. You just need to look for it. The downside, like I mentioned about the seedlings, is the genetic variation. There is a paper, if you want to find out, this is the year and the information. People in Colombia, another thing is always like, because people tell you, because you have read it. This is actually was one of the first studies that I found very interesting, because they look over 250 40 seedlings of Hass effect of the seedlings on 20 phenotypic traits. So they have seedling rootstocks and they look the phenotypic traits of Hass. And they found that the rootstocks have a significant effect in a lot of these traits, but especially, for example, fruits and yield. And this is what Mario Arpay and other people found before in California when they identified this Martin Grande rootstock, that it was very resistant, but when they put it into the soil, no fruits. So this is another example of what a progeny or seedling progeny, so you can understand why is this complexity more, like in the more visual way. This is, for example, your mommy here is PP35, is a Mexican and Guatemalan hybrid. This is just a phenotypic score for salinity damage, okay? 0 is the best, 5, the plant is dead. We have here around 150 seedlings, okay? Seedlings from the same mommy tree, which is PP35. And you see from this 150, your mother phenotype is right here in this score. But then you have some that are very, very good for salinity and those that are very bad. And you have all the spectrum from all this material. So you can see that individuals are totally different phenotypic. And this is some of the pictures. You have pictures of plants who don't have any symptoms of soil damage. You have intermediate, and you have plants that they have a lot of symptoms. And they are all seedlings from the same mommy, from the same tree, not even from different trees of PP35 clones. From one single tree, you can see this variability on the seedlings, okay? And this is another example from another West Indian and Guatemala. And again, you have these different seedlings and these different variations. And what I want to point out here is also what I was telling you, the West Indian giving you more salinity resistance. Look at the scores. In the scores here with PP35, which is moderate tolerant because it's a Mexican and Guatemalan, and we have the Guatemalan component that is bringing a little bit the salinity, your score goes from 0, which is the best, to 4, which is the worst. When you analyze the progeny of a West Indian Guatemalan mammy, your score shifts to the more resistant or less damage. Because you have a Guatemalan, you have a mother who is Guatemalan, and West Indian, which are supposedly the two best donors for salinity resistance. Okay, so this is when you can start looking what combinations you probably wanted to be for the rootstock. That's phenotypically. Now let's do genotypically. So you can see why the seedlings are so complicated and so heterogeneous. You have seen the phenotype. Now let's look at the genotype. If we look at those individuals, in this case, I think we look 700, I don't remember, but we have, these are the daddies. We do a parental analysis and we can find the daddies. This is like a CSI. You look at the kids and say, okay, who's the daddy? Because we know the mom. So you can see that around 160 of these 700 are self. So it's mommy and daddy, so PP35 self and give origin to these ones. And then these are the outcrossing. And we have 18 different parent donors for this population. And if you look at the whole genetic structure of the PP35s, all of these guys are Mexicans pure or Mexican Guatemalan hybrids. If you look at the population in this panel, we have Guatemalans, Mexicans, and the hybrids. So this is just to give you an idea of why the seedling rootstocks are heterogeneous genetically, and also that will involve phenotypical. Okay? What about the clonal? What's the problem with the clonals? It's the time consuming to make those trees. That one is very expensive. So Broca, for example, uses a method which is called frolic. That what it is, I can go through the whole process, but in general, you graft the rootstock onto the north seed. And then when you root the rootstock, you graft the xylem. But in the frolic method, you never separate or remove the mother seed. They put a ring, you strangulate the seed, and they separate the vascular system, so then you let the seed die because they kill the mother seed by blocking any contact with the rootstock part, so it doesn't receive anything. So eventually, by putting this ring, that if you guys receive trees from Broca, you will see a ring in the mother seed, in the north seed. This will be the connections between any flow of phloem, sap, or silent water between both. So you will have the roots of the rootstock and your rootstock and the husk. The microclonal is a method that was found in another nursery by Ernst. Unfortunately, he died a couple of years ago. So sad, he's from South Africa. And he developed a method which is a macroclonal method. The only difference, there are several different, but you still have a mother seed, as you can see here. But they made a different procedure that they will make the rootstocks root the rootstocks, and then they will completely detach from the mother source, the mother nurse. And what they will send you is this. So they will send you actually the rootstock in their own roots. You won't have the nurse roots. And people will say, but what is that the mother? Because sometimes if you receive fields of 2 years old plants to plant it in the field, all what you see is actually the roots that are leftovers of the seed, because the seed will still have nutrients and you will still have roots alive. But your rootstock doesn't have roots. So you plant your tree and a year later, your tree died. Or months later, your tree died. Why? Because the only thing that you have is the roots on the north seed, not the rootstock. Okay? So that's what is important. And the other method that is trying to people, especially industries, are trying to do is tissue culture. They want to propagate avocados or tissue culture. One of the persons who is the pioneer, and I think in my opinion, the expert is Nina Mitter, which is in Australia, and she has developed this technology, and she has done successfully for Reed and Belvic. And she actually was able to put these baby plants on tissue culture, let them root, and put it in the field. And she did some evaluation and see that a reed tissue culture grafted with has versus a seedling with has, that she didn't find any difference when at the time that she did it. I know a lot of nurseries, they say they can do this in tissue culture, they're doing this, but I don't see it. So now I'm like, I need to see to believe. The only thing that I know it works is this, because Nina already make it public, but I haven't seen a tissue culture in California yet. So the time that I see a tissue culture of California from any nursery that they are making it, I will show the picture. But I hope that happens. So, what is available in California is those, I think I need to go faster because that's my problem. I get excited and it's too forever. So, the clonal rootstocks, those are available commercially in California. There are different, so Westphalia have lots of them. They have new ones like Leola and Serrala. If you are interested, you can order it from Boca. You want to test one of these clonal rootstocks. They have new West Indians, hybrids, BCA01, that now they call them Tammy, Miriam, Benjacot. They have one from South Africa, which is Bounty, but it's in Quarantina. And there are also seedlings like Delvania and Ashdod, Walding that is used here, and Lula that is used also here. But we don't use, for example, Walding or Lula as a rootstock, even clonals in California. I wanted to start trying Lula because they have a very good resistance to salinity, so I'm curious about it, and it's a 3 hybrid mix. So I'll see. So moving gears about the introduction about, so what you know about how we're looking on the rootstocks breeding, something who can grow as a seedling, something that can go clonal. Our main focus, as I mentioned, was Phytophthora rubra. Phytophthora is how myci is have a 2 mating ties, A1 and A2. They have all the possibility to do sexual recombination, but as in many Phytophthora cinnamomum doesn't do sexual recombination, especially the population in avocado is a clonal population. What is interesting about cinnamomum is that it's a very invasive. It's originated in Asia, and it can attack 5,000 plant species, from trees, from Arabidopsis, from fruits. It just doesn't have anything, can eat everything. How we work with this, and I like to work since I studied plant pathology, is an integrative pest management. One tool is not enough. You can have the best rootstock, but pathogens are smart. Pathogens will change, and it will break down the resistance. So you need to get several weapons to the system to control the disease and not put pathogen pressure. That means chemical, not only chemical. So you need to combine the host resistance with chemical application if needed, and cultural practices. I told the growers, if you proper irrigate your trees, if you know your soil, and if you know that you have Phytophthora, you manage. If you know you have Phytophthora and you will plant, you're not going to put a very susceptible rootstock on the end of the hill, which is clay, because it will kill your tree. So you need to know your soil, need to know that you have the pathogen, and start raising beds. Don't, if you have a Phytophthora problem, just make a bed, raise the tree, and don't put it on put mulch. Mulch is very effective. Why? Because mulching attracts a lot of microbiome, a lot of beneficial organisms that will help to destroy the pathogen. Not completely, but it will help. Why? Because myci are different of fungi. If you students study, and if you remember your plant pathology, have it's more like a plant, doesn't have chitin, it has cellulose in the cell wall. So a lot of these pathogens, when you put mulch, they degrade them because they produce cell wall degrading enzymes. So they can destroy the cell wall carbohydrate component of the tissue, but also at the same can target the cell wall of the pathogens. So that's why the mulching is effective in Phytophthora. What's the problem? For me as a breeder, that my first goal will be Phytophthora, I need to know my threat. I need to know how my pathogen it is, how virulent it is, how complicated it is, genetical and phenotypical. So when I start my job, They did the first genotypic characterization of the pathogen population, and it was interesting that at that time, Paglasha with Greg Duhan found these two groups of Phytophthora cinnamomum associated to avocado, it's all avocado. And you can see the blue one is a clay of very specific genotypes and some phenotypes that we found later that they were only found in San Diego and Riverside. San Diego and Riverside is close to Mexico, you know, and then there is a lot of more trade probably there that is more like informal. So one of the hypotheses that maybe this group, because you couldn't find any clay in other parts, just San Diego and Riverside. So then one of the hypotheses that that clay was introduced from Mexico. Then I read some papers of Ochoa, Salvador, and they also find two clays in avocado in Mexico, Michoacan. So the interesting is that this clay in the southern growing areas, if you mix it and inoculate at that time the rootstocks available, DUSA, the susceptible rootstock that we use as a control, DUSA was released because of resistance. And this is a perfect example that I get the growers. Because growers think that if you have one rootstock that is resistant, it will be resistant all their life. It's not true. This is the perfect example. DUSA and Thomas were resistant from the Westphalia, South Africa, and Thomas from the UCR like years ago. It's with one population that was 10 years ago. Now, you see with the clay one, with this group of isolates, the resistance still hold with the control. But when you use a mix of more virulent isolates, look, your DUSA have less root health, so it's more becoming like this, like you're susceptible, and like Thomas. And then the new rootstocks who were selected with actually isolates from these areas, they're holding resistance. So you have to keep changing, because pathogen change, so you have to change. That's why it's always complicated. So how you do that? We tested the hypothesis that this group come from Mexico. So my student, Naida, who now he graduated last year, we work with Silvia Fernandez from Michoacan, and they collect and give us a lot of Mexican isolates. And what you hear, just look at the color, because this is the most simple way to look at it. The Mexican isolates are the purple. Fuchsia. The purple are the California southern growing areas. These orange are northern areas, what we call them Santa Barbara, Ventura. You only have in those 50 isolates 2 Mexicans, and the rest of their isolates from Mexico, Michoacan, were in clay 2, grouped with the California, the Southern California population. So that tells us that this, indeed, Mexico might have been the source of this new clay that appeared in California. Now, we cannot trace back to see, when this time occurred or what happened, or if the isolates who came here can already be resistant or to more virulence, we don't know. We have to do more study, but at least we answered the question. And the other thing that we found in our study is that the A2 clay isolates are moving regions, because before it was only found in San Diego and Riverside. And now we can find this clay too in Santa Barbara. So there are different ways how the isolate can move. It can move to the pickers, it can move through the chipness of rootstocks. And then these isolates also have different phenotypic. There are more virulent. Some of them are more insensitive to potassium phosphate. Potassium phosphate is the major fungicide, and it has sense because if your pathogen is more virulent, you need to apply more for potassium phosphate, and then you make the population more insensitive, like the antibiotic. You take the same antibiotic, you get resistant. The same happened with Phytophthora. These are some of the UCR rootstocks grafted with HAS and tested for resistance to this using this population, and you can see that DUSA is succumb or is breaking down the resistance with this population, but our isolates, because they were screened with this, still hold it. They also then do California, and we cannot wanna see because our rootstock have to be hopefully everywhere. So how is the population in other states? So we have different states, Florida, Hawaii, Oregon, and we found is that this Phytophthora cinnamomai, remember, is clonal. They don't do a sexual reproduction. So the genetic diversity is not very big, but they have a great adaptive capacity to make the resistant rootstocks susceptible, to make the potassium phosphate insensitive, and now they can grow to different temperatures. Usually they grow happy at 25, but then some isolates from California south, which is the hotter areas, some in Florida and some in Hawaii, can even grow at 32 degrees. And the optimal temperature switch to 28. So there is a local adaptation. So the pathogen invade, start new area, and they adapt. And this is a potassium phosphide, and you can see the big variability. You have highly insensitive isolates with 900 EC50, which is mean the amount of chemical that you need to use to inhibit half of the growth of the mycelium. You have isolates that have 800 micrograms per ml and isolates that are less than 50. So, and then the highest one are California, of course, because California growers just really use lots of potassium phosphide. And there are some from Florida. So this is where the integrated pest managers play a role. It's like you can see, if you use a susceptible rootstock, potassium phosphate or moderate resistant, you see there is no effect of potassium phosphide with the control who don't have any fungicide. Different, they have the C letter chair and the B chair letter, so there are no difference. But in potassium phosphide, but when you use a moderate resistant or a resistant rootstock to Phytophthora, now your potassium phosphide is different from if you don't use. So potassium phosphide is still used, but if you use it with a resistant rootstock. So that's why I told the growers, you need to use, if you're going to use potassium phosphide, use it with resistant and rotate it with, for example, Orondis. Orondis is oxatiopiperine, who has been registered in California and is very effective to control root rot. This is Phytophthora root rot incidence, and it's very effective, and now people have to combine potassium phosphide with Orondis. So what we do also is a selection of different sorts of seeds. That's how we do our selection. So we take the seedlings from mothers who have some kind of resistance to Phytophthora, they have salinity resistant too. So we screen them, and then we screen them for Phytophthora resistant. You can see here 0% after the screening. So all the roots are dead. And this is a highly resistant selection, which have pretty much very low damage. Visually, and then we collect pathogen and we do all recoveries to be sure that the pathogen actually is infected and needs to escape. We re-inoculate the plants before moving it to our germ plants. But you see, we have to screen 3,000 seeds to get 50. This is the space of the whole greenhouse we have to use. So we need to move to a more molecular techniques to have genomic selection, a marker who allows to get these resistant areas in the chromosome, that we don't have to do all this inoculation and just focus on the ones who have the marker. So we were able to do some kind of genome-wide association studies with these mapping populations of these segregated progeny from the PP35 mother, and we were able to identify one SNP marker located in chromosome 6, who is associated with that resistance on the mother. And the most interesting thing is when we look at, we have a lot of RNA-seq data from avocado, from other hosts, and when we look at this SNP area in the chromosome 6, we found a candidate gene or genes family in that area, which is associated with a resistant plant, which makes us more confident that now we can try to validate this marker and this candidate gene and see if we can, rather than do all this inoculation of these 3,000 seedlings, we just do a PCR and find the presence of this gene or this marker, and then from 3,000 only inoculate 20, for example. So we don't have to use the whole greenhouse, but maybe one bench. So that's the goal of trying to use genetics to facilitate the breeding. We, of course, need to do with those 25 materials, we need to do the clonal screening, because sometimes in the seedling you can have escapes. And also something that we do in the program is we put material that it is already available. So we know that our resistance, because we need to at least set our material. So our resistance has to be similar or more than what is available and have other characteristics. So when we do our screening, we still put DUSA, and we still put one of these one is Leola and Serala, which is the new South Africans who have good resistance. And then we choose seedlings. For example, this is inoculated with the more virulent isolate we have, and with a mix of all isolates representing the whole population in California. And then you can see that there are some like this GD6 or like this PPs at 102, which is my selection. You see that they're very good comparing with DUSA and the susceptible rootstock. So then those ones will be moved to other traits. And if we look at the whole story, we ended up now, currently we have 224 of these selections for Phytophthora root rot resistance, which are unique because we genotype them all and we know there are different things. We have in general, the germplasm is 300 unique accessions with 70 international material, and we have 14 per se species. So we're using all this material, we divided by tiers. So the 5 rootstocks that we're releasing 35 and 40 in this fall, we have 2 in the pipeline, 42 and 45 and 80. What we have at this long term of the release on years is because we need field data. You can have your trees, all the data in the greenhouse showing you that is perfect, that is resistant, that is salinity increasing. But if you put it in the field with HAS or other scion, it needs to produce. Because it's no work. I mean, it works scientifically, but not for the grower. Because the grower wants something that is good, but it produces. That's why once you get to the stage, it takes at least 10 or 20 years, not 10, because you need at least 3 years until the first production, another 3 or 5 years of productions to be sure that your tree produces in all different conditions, and then you release it. So let me go skip to the most interesting part. So this is the PP40. I want to show this one because some of you guys are doing PP40, and some of those talk with Carlos, who grows here, have a growing in California with lots of orchards, and he's using Torocanio. Torocanio is a Mexican Guatemala who have a moderate resistance to Phytophthora, but it has a very good salinity. Could be some Mexican than Guatemala. PP40, which is the new selection that we are releasing, is a seedling of Torocanio. And it's segregating their salinity resistance, but the resistance to Phytophthora is higher. So it's like when you have kids, your kids, or when I have my students, they say, you have to be better than me when you graduate. And this is the case for the PP40. And Estedon, which is already released, is their Torocanio's childs, and they have Phytophthora resistant enhanced, and they keep the salinity. That's what they're still using in California for salinity and for Phytophthora. And you can see here in the greenhouse, Thomas is very susceptible to salinity, and PP40, after 5 months in a 1.3, this is the salinity treatment, 3 EC and 600 ppm of chloride. And to give you an idea, avocado starts losing production at 100 ppm of sodium and chloride. And we keep our material in like 600 ppm of chloride for like 6 months, or 195 of sodium for almost 6 months. And this is only one score. PP40, comparing with the susceptible Thomas, it doesn't show that much tree burn. You can see here the picture after 5 months. We have to stop the experiment because Thomas was dying. And we did a lot of stuff, but one of the things that we did is we want to see the sodium and the chloride contained on the leaf and in the roots. And what you see is that in both of them don't vary on the amount of sodium and chloride that they absorb from the roots. But what it makes different is that in the leaf, which is when you're going to see the leaf damage, you see that Thomas, which is the susceptible, have way more sodium than the PP40 and chloride. So we know that PP40, because this is ungrafted, this is the PP40 alone. So PP40 is not excluded, but it's a sequester. So it cannot take the sodium and the chloride, but it just keep it sequestered in the leaf. And we're working with experts in UC Davis, and they're doing some anatomical sodium distributions, and then it seems very interesting. I don't understand the percent because it's too complicated. And that I haven't get yet to understand the anatomy. But just to finish up, this is just what I was mentioning. All this research pick up our best candidates for the rootstocks, but we cannot release if we don't have field data. So we have, currently we have 19 plots. When we have, you can see all over California, all the way from Santa Paula, Santa Barbara, and San Luis Obispo. We have it under different conditions. Some of them have Phytophthora, some of them don't have Phytophthora, some of them have all. So it's like the more variation that we can have, the type of soil is different. We evaluate everything, especially yield. And then we have now rootstock scion trials. We have one here in the station with Bruce and Jonathan. We have our 5 PPs with different scions, and we're trying to see how this one behaves in California and produce in Florida. We have one in Texas, in Hawaii, in Puerto Rico. We have our material in China, Australia, India, Mexico, so South America. So hopefully they can also, I was talking with the Mexican partner who have the material. They're very happy with the PP80s and the PP45s, and the PP80 also looks good here. So who knows? So this is just a couple of examples of the ones who have really high soil. So you can see this is a rootstock scion trial that is the replication from here. It was established 2022. The EC of conductivity for salinity is 2.4. Chloride is almost 259. pH 8.7. You can see the pH is super alkaline. They have a lot of calcium carbonate. Soil you have from loamy sand, you have sandy loam, you have in Spear Miller, you have clay soils. So we have all this grow or tested in the orchard farmers, by the farmer, and getting the production data, which is the most important. So this is an example of the production data. And this is, I point this one so you guys know what I was talking about, the PP35. The PP35, depending on the environment, made the tree or has a small. You can see here, PP35 versus PP40 in this plot when they have high salinity and chloride. And you can see that it's half of canopy of the PP40, but it's very vigorous, very nice tree. Look at the production over the years since 2022. In total fruits and yield, it overgo more than PP40, even having the same canopy. When you look at for tree, also it's similar. There's no big difference. Look at Dusa. So when we analyze this, we have a small tree. It doesn't mean bad. You have to see the tree in the whole context. That's what we want. We want a tree who is highly productive, but doesn't make the tree too big. And this is the problem when you have only a West Indian rootstock, because West Indians tend to make the scions big. And California, at least Californian grower, doesn't like big trees because it's too expensive. So this is what we're looking. This is another example when PP80 that I wanted to show the productivity in this is really good under salinity, but we cannot release 80 yet because we don't have that much production data on this one. Only this is the first year for that. So this is the rootstock scion trial, and this is what I wanted to show. This is the rootstock, the replicated trial that I was telling you when we were looking the one in Florida. This is the harvest of Haas and Jen on the 3 rootstocks, 35, 40, 80, which this we decided what to plant because we know this field has problem with salinity, so we don't want to put 45, which is sensitive to salinity. But you can see here that with the gem, the best ones are 35 and 80. And if you look at in the Haas, Also, Dusa is with 40 and Tolocano. So this is the first data that we have in the trial, and I was saying we have more than 100 in some cases fruits per tree on some of this material. So we're very happy. So again, Jen, PP 35 and 80, and then this is 35 and 40 and 80. That seems no difference. And because, I mean, I think that's an, so this is in general production data, which is what we care. You can see that our rootstocks are better or equally to DUSA, which is our standard. So we always have these standards on terms of production, which is what I'm showing because that's what is important for the growers and for us. Mortality is also important in this field. For example, there are some trees that they have 50% mortality, other trees don't show any mortality in the plot. It's important to look at the mortality. This is a productivity when we have a lot of the commercial rootstocks and we have some of the new generation rootstocks like GDs are the best producers in this field. So then we can move and test more of these rootstocks. And then this is just a characteristic of what is going to be released based on these 19 tests on different areas in California and the different conditions. So we have a lot of calcium carbonate, 350 on the field of chloride, high pH up to 9. So it's high Kalisa. And this is my take-home message that I want to finish with this. Diversify. That's what I told growers. You have a lot of different type of science that it works for your local area, for your orchards. You have different rootstocks that it works. So if you plant all Dusa, and if Rosellinia came here, Dusa is very susceptible to Rosellinia. All your orchard is in Dusa, all your orchard is gone. So, and then if you have a field that is clay and you plant on Dusa, and Dusa doesn't like clay soils, all your orchard is gone. So you can plant a patch of Dusas, if you don't have clay problem, a patch of Tolocano, because you know all of them produce with husk. So you are not going blindness. So diversify, and then you will keep your orchard healthy. Irrigation is very important for controlling disease. Then you will have a healthy orchard. It doesn't get sick. It's like us. If we are not stressed, which is impossible, and if we are very well healthy and eating properly, we're not going to have more healthy problems than the people who don't take care of themselves. So keep your orchard happy, healthy, and you will have less problems of diseases. And this is where my team showed out, but a lot of work has been done for my students, my postdocs, the breeder who just finished in my lab. Now I only have Amber and Matthew, which are my assistant for the field. Matthew started his PhD last year, and Ben, who is my PhD. So from all these people, I'm back to 3. So I'm on sabbatic, so I downside my life, so trying to have some money, and then Bill again, but I just wanted to thank all these people who are very instrumental. From undergraduates, he is an undergraduate who worked in the program for like 3 or 4 years till he graduated. Now he's in medical school. But yeah, so thank you. If you have any questions, sorry it went too long, but.
But maybe just 2 questions. Yeah, somebody has a question. Yeah, for him.
Yeah, thank you, Patty, for your seminar. I think it was very exciting, and thanks for your work that you presented to us. And I was particularly interested about the invasive history of the Phytophthora cinnamomum. I want to ask you what kind of marker do you use to find those clades, and also what is I saw you. Maybe, I don't know if you mentioned it, maybe you did, but you had a map of Southeast Asia, maybe. What's the story behind those? Because I understand that only the mating type A2 is exclusive to avocado. And what's up with the other A1?
That's a question that I'm very interested because so far, with having been an A1 associated with avocado, so there are a paper who mentioned that they use an A1 from avocado, but in California, a long time ago, they report an A1 avocado, but it was wrong. Because currently all the population that I'm seeing, even with my colleagues who have previous studies and they publish the populations on avocado, all are A2. Doesn't mean that the A1 cannot infect avocado. The A1s can infect. My coffee did those experiments and John Mangi. They can take A1 from camellia and then infect it. They can do sexual reproduce because you mating time with A1 and A2 in the lab. It's just in nature, it doesn't happen, you know? There were some sexual recombination found in South Africa and in Asia. Regarding the movement, I cannot claim that much, and that was actually the reviewers, because we only have a few isolates from Mexico and Asia. But definitely, there were two introductions from Asia in California. One was from Papua New Guinea, and the other one was from Taiwan, which are the two centers of origin. Now, we only have 5 isolates from Asia because we get it from the collection. We need to increase the isolates from Asia. But the nice thing is, before us, Matteo Gambellotto, you guys know him. Matteo Gambellotto works in Phytophthora cinnamoma, but more in forest. So he published before a very big paper with having a lot of A1, A2 from avocado, from Mexico, from a lot of places, and also a lot of hosts. And he has a lot of Asian material, and he also report that California went from isolates from Papua New Guinea, and Taiwan came to California, and that Mexico also have introduced, that they have been exchanged between California and Mexican isolates on Phytophthora cinnamoma. So that's actually, I need to resubmit my reviews, and that's what my point. I'm getting this paper. Yeah, we only have 5 isolates, but we found in the same that Matteo found with more Asian isolates. So I think it happens from Asia.
Yeah.
Yes.
This is a more general question. It sounds right, of course, almost always that diversifying is the way to go. But sometimes, us as consumers, the market is looking for very particular, almost general way of, traits in the fruit that are sometimes similar. So how you consider those, conditions within your breeding program, considering that people sometimes are not that eager to test, diverse products, but rather like the same kind of,
I think I got it easy because I work with the rootstocks. So I don't breed for this fruit for what you're saying. And this is a big, that's something that I understand because it's a lot of consumer and in the science part, you want to keep something that the grower, the people or the public wants to eat.
Yeah.
But, at the end of the day, I always go back to the history of Fuerte in California. Before Haas, it was all about Fuerte. I wasn't there, I probably wasn't even born, but that's what I learned from a lot of people like scientists in avocado, like Marilu and growers there. Before it was only Fuerte, and everybody was happy with Fuerte, until they found Haas. And why it was the nice Haas is that it's brown or it's black when it matures. So that is the key how they marketing. They put a lot of money on marketing, and they switched the mentality of the people in California from a green skin to black skin avocado. But Haas wasn't, like Fuerte wasn't bad, Fuerte is good. So I think if I'm not a scion breeder, but it is important because the worst epidemics happens because monoculture. That goes back to, I also work with Phytophthora infestan, which is the late blight. And if many remember, is the potato famish Irish famine. Why it happened? Because you only grow one type of potato. What Phytophthora exotic bad? Laura will has susceptible Laura will bad. So I think it's all about trying to get the scion breeding, so the breeding on the scion, they need to get something that people like, something close to Haas, or something who do a lot of taste panel to have this good combination of fat, flavor that people like, and then market. The same way that you market Haas and may change people from Fuerte to Haas, why not find another green skin that is so good, but it's also resistant to laurel, but it's good. And you make taste of partners, people like it, it's acceptable in the public, and then you market, and then you get the market. It's all about publicity. If you don't public your products, people don't know and don't get interested. And mine it works because how you sell it. So I think, but bottom line, I think genetic diversity is the key for like, don't die.
So thank you, Patty.
Hey.